I just ran across this about the inventor of the tests they're using for COVID. He tells you it can't detect if you're sick. Notice how they remove the video because it's truth. Now, I don't know what video she's referring to, but the article she sent has nothing to do with proof of people dying from the vaccine. They aren't lawsuit documents. It's merely an article which says PCR tests can't tell you if you are actually ill with something. And we know that. And that has nothing to do with COVID. PCR tests are designed to tell you, is a particular signal present in a swab? You might physically have some virus particles in you and you might not actually be sick. That's not news of any kind. And so.
Wow. So David Packing here just basically debunked Dr. Samantha Bailey's entire thesis of her truth about PCR. Hi, everybody. Just welcome back. This is part 2 of my debunking of the truth about PCR. A little bit of background here. So part 1 is located up on my page. I'm realizing this is probably not going to get a lot of likes, and I really don't care. My purpose is to just put this out so that the information is out there. And I'm kind of learning as I go about, turning off notifications so that it doesn't pling in the background, and bringing the microphone closer so that people can hear me a lot better. And also, keeping it a little bit shorter in terms of the rants that I'm doing. But, I welcome you to take a look at this. It's probably going to be directed more towards people who are already in my corner, who can further process this, but maybe don't have time to get all the technical details out. So I just want this to be easily accessible, and a point by point, just on exactly what she's saying, because she's saying some pretty crazy stuff. So again, here's the video. This stuff about PCR, it's rampant, and is not going to stop. So here we go.
2020 seemed to be the year when PCR tests became a household name, riding on the wave of...
The last time PCR tests were a household name was back in the 90s during the O.J. Simpson trial, because that was when PCR testing for forensics came into the main fray as something that you could use for forensics. And it's amazing because she says at the end, PCR tests are great, forensics are one application, and then goes through an entire homage about how great the inventor of PCR, Kerry Mullis, was, totally ignoring the fact that he threw PCR under the bus on forensics. So I don't know what application she would consider okay for PCR, or why she explained, or how she can explain that application is okay for PCR, but this application for diagnosing disease is not okay for PCR. There's no technical difference between those processes.
Of corona mania. Prior to this, PCR tests were far less well known to the public, and even much of the medical community had little engagement with them. Now it seems...
that's if you've not done anything in a laboratory, I guess you would have very little engagement on PCR. If you run a laboratory or near a diagnostic lab, or if you're a medical doctor diagnosing things, you definitely for sure know that PCR is a thing. I don't know what she's talking about. The only thing I can think of is she doesn't actually practice medicine, so she doesn't actually know what tests are being done. She mentions hemoglobin tests later. This is the basic of basic primary tests, and she doesn't go into anything that's even more complicated than that.
Everyone has something to say about them. However, like my recent video exploring what exactly is a COVID-19 case, it is useful to go back to the basic science to understand what the foundations are, so we can see where it can lead and often where it can go wrong. So what is PCR? PCR stands for polymerase chain reaction. Polymerase means an enzyme that catalyzes the formation of a polymer or long chain molecule from smaller units. In this case, we are talking about DNA, the molecule that carries the vital genetic information for much of the life on our planet. Chain reaction means that the process is carried out multiple times in sequence. This amplification...
Chain reaction means that it's a small individual reaction that sets off a larger reaction within sequence. So she got it right that I guess it's in sequence, but then she totally leaves out that it causes chain amplification of the DNA. I mean, she does mention later that it does amplification, but the fact that it is a chain reaction means that it starts off with just one tiny reaction, and then that amplifies into many, many, many different reactions.
Process means a minute sample of DNA is multiplied millions of times to produce sufficient material to allow for genetic and molecular analysis.
Terrible gel. Oh my gosh, this is awful. I would never accept this in lab. Look at how fuzzy these bands are.
The history of PCR started in 1976 with the discovery of...
Okay, so in her other video, the history of PCR started with the invention of PCR by Mullis, and she goes on and lionizes Mullis, totally putting him in a total vacuum with no other colleagues that worked with him on it, and he's done absolutely everything, including the work that was done on Taq polymerase, which is not true. But now it suddenly is starting with Taq polymerase, but PCR predates use of Taq polymerase. In Mullis's paper, which is right here, you can see that they were in fact using an E. coli, Escherichia coli derived polymerase, polymerase 1, not Taq polymerase. In fact, Taq polymerase was first used later by the Cetus Corporation. Now it is mentioned that Mullis actually suggested the use of Taq polymerase as a thermal stable polymerase to use for PCR. But if you notice, Mullis is gone, and he's not in this list of authors when they used the Thermus aquaticus DNA polymerase for their 5' to 3' endonuclease activity that allows for, oh, by the way, DNA probing, which he leaves out after her very simplistic explanation of how PCR works. So, she's like, I'm not going to go into the details, but I'm going to leave out these details that also show how PCR is a very, very sensitive, very specific type of test.
Of Taq DNA polymerase. The enzyme was found in Thermus aquaticus, a bacteria discovered living in hot springs in Yellowstone National Park. Because the enzyme could withstand temperatures of 95 degrees centigrade, it was a practical molecule for the thermally dependent PCR process. Prior to this, fresh enzyme would have to be added at every single cycle. Things really took off in the 1980s when Mullis realized how to put Taq polymerase to use, and the enzyme became commercial.
Nope. Mullis on the way out the door suggested Taq polymerase would be a good enzyme to use for commercial purposes. And then he left, and his colleagues at Cetus developed it into a further method using probes to do the PCR. Nope, he did not work with Taq polymerase at all.
Commercially promoted. Mullis was awarded the Nobel Prize in Chemistry in 1993.
So again, part one of my debunking, which actually came after this, she lionizes Mullis as this great scientist, ignoring the fact that he's done absolutely nothing after his invention of PCR, and goes into some other details about how PCR functions that are incorrect. So check that video out and let me know. It's the first one I made, so it's a little bit rough.
And is generally considered to be the inventor of PCR.
I won't get into all the technical details of PCR, as for our purposes,
especially the technical details that show that PCR is a very sensitive technique, because that goes against her mind frame that, oh, you can't use PCR as a diagnostic test, it's not sensitive enough, it's not specific enough.
Principles and application are more important than all the biochemistry. However, for those of you interested, I'll include links with more detail below in the description.
In summary, the process starts with heating the sample of DNA. This separates this double-stranded DNA into two pieces of single-stranded DNA. Primers are then added. These are short pieces of DNA that are designed to flank the target section of DNA for amplification. Usually 2 primers are used, one to bind to each of the opposite strands of the target DNA section. The polymerase then synthesizes a new DNA strand that is complementary to the target DNA section. It builds this from nucleosides present in the reaction mixture. This essentially completes a cycle, and the amount of target DNA has been doubled. The cycle then starts again, but this time with around twice as much target DNA as the first time. The DNA Learning Center had produced a nice little video outlining the PCR process in 3D, which you can check out in the link if you're interested. The cycle is typically repeated 25 to 40 times, and it is important to realise the exponential amplification of material that occurs with these numbers. For example, at 40 cycles, there would be around 1,000 billion copies produced. However, Taq DNA polymerase has a certain number of mismatch errors, so higher cycle numbers are more likely to produce inaccurate results. It is also important to realise that a typical PCR test only detects a small sequence of genetic material. It does not detect anything close to the entire genome of a purported organism.
Okay, so here she leaves out that the PCR uses probes, which is a big point because you have primers which are very specific, and I will go in and show you how specific those primers are in a second. And then you have a probe that probes in the middle. So in order for the PCR probe to work, and this is a great article by McGill University that is highlighting how this works in terms of like a book. So if you think of an entire library of books, and then you think of just this one book, this one phrase from this one book in Sherlock Holmes, right? The chances of you finding this exact phrase, unless it's paraphrased in another book, is extremely low. If to find these words in this order, and that was how, and then a second primer going in the reverse direction, a woman's wit, to find those in order are pretty low. You could probably find and that was how in another book, in another section, in another paragraph. But for the PCR to work, it needs to be within a certain range of the reverse primer, a woman's wit. So if you don't have both of those together, then you're not going to be able to form the PCR product to induce the chain reaction, right? And additionally to that, to make it even more specific, in order for that product to be registered in the reverse transcript, in the real-time PCR, it needs to be detected by a probe. So the probe needs to latch on to an exact phrase that falls within those two primers. So now you have three levels, a forward primer, a reverse primer, and a probe that needs to attach to that specific sequence. So you have three levels of specificity within a single reaction. And most of these SARS-CoV-2 detections for diagnostic tests on COVID-19 are multiplex reactions that are run 3 times, right? So one detecting the RNA-dependent RNA polymerase specific for SARS-CoV-2, one detecting the envelope protein gene for SARS-CoV-2, and one for the nuclear protein for SARS-CoV-2. So that's 3 levels for 3 different reactions of specificity. And then when she goes in and very simplifyingly says, oh, it only detects a little piece of a little section of the genome. It's like in any other context, in a genome for, say, wheat or humans, where the genome is huge, that would be still a very specific test. In the context of COVID-19, SARS-CoV-2, where the genome is 29,000 base pairs long, that's a significant portion of the genome that you're detecting. You're detecting it in 3 separate places.
Okay, so we have a pretty neat way of detecting tiny amounts of DNA and actually RNA as well. What could possibly go wrong? Well, quite a bit when the technique is used to draw conclusions beyond that for which it was designed.
If you're not designing the PCR reaction in lab correctly, a lot can go on. If you're an undergraduate who has no training in it, and this is your first time doing it, we've been doing PCR for 35 years. Regulatory agencies have been monitoring and regulating and allowing use for PCR on diagnostics for 35 years, because she leaves out that Carrie Mullis worked at CETUS to design the PCR test to test for sickle cell anemia as a diagnostic test. It started out as a diagnostic test. So So to say that, you can design PCR wrong, you can, but if not for the purposes that were intended for testing for COVID-19, because we've been through these ropes for 35 years. It's not unknown.
The inventor of PCR himself warned that the PCR test doesn't tell you that you are sick. These tests cannot detect free infectious viruses at all. And as I've discussed...
And let's just go back because David Pakman said it the best.
What video she's referring to, but the article she sent has nothing to do with proof of people dying from the vaccine. They aren't lawsuit documents. It's merely an article which says PCR tests can't tell you if you are actually ill with something. And we know that. And that has nothing to do with COVID. PCR tests are designed to tell you, is a particular signal present in a swab? You might physically have some virus particles in you and you might not actually be sick. That's not news of any kind. And so I wrote back at this point and I said,
there you go. Even David Pakman, who does not have a molecular biological background, can recognize that the purpose of the test is to test for the presence of the viral fragments that can cause disease. There are asymptomatic people that, there's an asymptomatic part of the disease that can spread it. And then there are asymptomatic people who don't develop symptoms or developed extremely mild symptoms who can still spread it.
In previous videos, the manufacturers of PCR tests usually have disclaimers.
Okay, so now this is the first RT-PCR SARS-CoV-2 test kit that she highlights that is specific for laboratory use, and then she points this out. She'll jump to other SARS-CoV-2 RT-PCR tests to make other points, never minding the fact that, okay, well, this is intended for, from the manufacturer itself, it says it's only intended for research purposes because they're doing research that requires detection of SARS-CoV-2. But never mind the fact that the FDA then comes in and says, okay, yeah, this is good. You can use this for diagnostic purposes. So she totally ignores that fact. And then this is like one of 4 kits that she's going to use to make her specific points. Ignoring the fact that, no, the FDA and other agencies around the world have their own kits, or they have their own internal diagnostic RT-PCR setup with specific reagents, specific primers, specific probes that they've then validated to say you can use this for diagnostic purposes. But just taking this for research use only totally out of context to say that no PCR can be used for diagnostic purpose. Totally disingenuous.
Stating that they are not suitable for diagnostic purposes and that they are non-specific. Let's explore.
She says that they are non-specific. The test that she picked right here, the Real Star SARS-CoV-2 RT-PCR, is very specific, and it says it right in the detailed notes that it doesn't detect other coronaviruses or other viruses. That is actually very specific. Now, she utilizes later a test that is not in production anymore by a very small diagnostic company that says that there's non-specific interference by other viruses. It's one test that was not used for clinical settings. It was used for research in clinical settings that I can't find any reference to. That's not FDA approved. So it hasn't contributed to the overall diagnostic testing, but she'll take it out of context and use it anyway.
Some of the reasons why this is so. Firstly, the very nature of the biological samples is problematic. When a biological specimen, for example a nasal swab, is taken from a living host, it contains all sorts of things, including genetic material from any number of microorganisms. Humans are covered in billions of microorganisms, most of them living in symbiosis with us, meaning they don't cause any harm or any disease. In fact, many of them are essential to our very existence. Experiments with newborn animals have revealed that attempting to raise them in sterile conditions leads to rapid death. Most microorganisms found in and around us have nothing to do with illness, so the mere apparent detection of their presence is not a cause for alarm.
Right, but we're not detecting all of the microorganisms that live around us and on us. And this PCR test, if you would pay attention to how specific it is detecting the microorganism that we know causes the problem. The argument that she's using here is that because we don't know every single sequence that's out there, never mind the fact that we've sequenced 60,000 at least genomes around the kingdoms of life, and have a pretty damn good idea about how those taxonomies of organisms have their genetic structure. You're not going to find these specific genes from this specific organism in other organisms. It's not gonna trigger it because they don't exist in that pattern, in that way. So it's a highly specific test for not just coronaviruses, but for SARS-CoV-2. And she totally ignores this. It's like it doesn't even exclude just coronaviruses. It excludes only down to this very specific viral species that's causing this respiratory disease itself. So this is a total nonsense argument. You're not going to find detection of any of these, and the clinical samples that have been taken as negative controls are absolute proof that this test is not going to just randomly pick up microbes.
Additionally, as Carrie Mullis pointed out, a PCR test does not detect an infectious agent. This is because it is an indirect test that only detects genetic fragments of organisms.
Let's hear it again from Pac-Man. He said it the best.
Our tests can't tell you if you are actually ill with something, and we know that. And that has nothing to do with COVID. PCR tests are designed to tell you, is a particular signal present in a swab? You might physically have some virus particles in you and you might not actually be sick. That's not news of any kind. And so I wrote...
So there you go. Same argument over.
Your body may have encountered a potential pathogen that your immune system rapidly destroyed, but the PCR test still detects remaining fragments.
If the PCR test, if it detected these small remaining fragments, there's only a very minute time frame that's actually possible. How do you know that the test is testing your degraded viral load because you're getting over a reaction versus a brand new viral load because you're just getting the virus? You can't tell that. So there's a lot going on with like PCR thresholds back and forth about how it's too high and it's too low and it's being manipulated by a global government cabal. But the answer is, like, if you're testing positive for it, you've come into contact with it, and so have the people you've been around. So they may not know. Though, you might catch it to where people have just been exposed. So if you, even though you tested really low at a high CT threshold for this virus, it might just be that the virus is new or that the virus is old, you're asymptomatic, and then those around you need to be notified because you could have spread it to them.
It also needs to be established that a genetic sequence is absolutely specific to a certain organism, and this is where diagnostic PCR can fall apart. Due to the almost incomprehensible amount of genetic material on the planet, it is rash to assume total specificity when it comes to particular sequences. What about SARS-CoV-2? Was it even formally isolated prior to the development of a PCR test? As you are aware, we are currently...
This is a crazy argument that she's precluding to. First of all, she's using scientific language against common language to say, like, have you specifically purified it right down to the to the pure essence, like you were going to use it for X-ray crystallography to look at co-proteins or the structure of it, and then saying like, oh well, they haven't even isolated it. When in my show notes here, you can see there are so many examples of isolation in the literature. It's identification and isolation from patients in Korea with COVID-19. Let's go to the second one. I believe that this was the first one that they isolated, and you can see right here that they isolated it from these patients. And the way that they did this is they took infectious material, they plated it out on clean cancerous lung tissues, and then from there we're able to view the viruses infecting it, and we're able to then isolate it in a buffered solution. So you can see here's a CT of the infectious patient that they took it from. Here is a transmission electron microscope photo of the infectious cells being infected. And here you can see a visualization of the SARS-CoV-2 virus, very specifically with the co-proteins right here. And then she goes to say, is like, well, have you isolated? Have you gotten away from all of this cellular debris? And it's like, no, I just had it either spun down and plated and viewed and said, hey, look, it's here. Or I have it within a cellular matrix that I can see it budding off of. And she's using this as false dichotomy to say like, well, then you don't know that it's there, and therefore you can't make a PCR test off of it, when you can absolutely just snag viral RNA, the entire genome, right from this. You don't even need it to be this purified to snag viral RNA from.
Working on the third English edition of Virus Mania, and this has been something we have been looking into. We asked the groups that reported that they had identified the novel virus by electron micrograph whether they had formally purified the virus.
So they identified it by electron micrograph. Have you purified it so that you can extract RNA and make a COVID test? No, because you don't need to do that. Here they've done it by a density band to show like, hey, we have EM images obtained from cellular culture. This in cellular culture, this causes a disease, and we used density gradients to separate those out, and we can see it right there. Have you purified it like you're going to do X-ray crystallography? No, because you don't have to.
The responses were all the same. No. Even Michael Law from the Robert Koch Institute wrote in an email that we received on September 4th, 2020, I am not aware of a paper which purified isolated SARS-CoV-2. Hence, it cannot be concluded with any confidence that the so-called COVID-19 PCR tests were specifically calibrated to a brand new viral pathogen.
Yes, you absolutely can. Because the genome is known. The genome has been known since January of last year. And the way that they isolated that genome is by taking it out of infectious individuals, plating it out on cell lines in culture, and then extracting viral RNA and getting that genome. And all you need is the sequence of the genome in order to make the qPCR test. I don't know why she thinks you need to have it into the super purified level like you're going to do X-ray crystallography. I think she probably knows this and probably worded her email so that the scientists would, in their way of being super specific and also her way of just leaving out the details, could say, oh no, that wasn't purified, so therefore we don't know that SARS-CoV-2 causes COVID, or that the PCR is not calibrated correctly. Whatever that means. It's not a calibration thing. That calibration comes later after you design the test, and all you need is the sequence of the genome, or even a part of the sequence of the genome, to make these tests. In fact, the CDC didn't even have their own isolates. They just took the viral genome that was produced from the Wuhan virus and replicated up a part of it to use as their positive controls. And that totally worked. Okay, now she's going to go into probably the most frustrating and annoying part of the talk that I have ever had to sit through. And I feel so bad because Barbara McClintock was an absolute unbelievable genius. And the way that she butchers, the way that she absolutely takes her knowledge and just warps it into what she considers science is disgusting.
The research work of geneticist Barbara McClintock is also worth a mention here. In her Nobel Prize speech from 1983, she reported that the genetic material of living beings can constantly alter by being hit by shock.
She was not talking about the genetic material in terms of DNA itself. She was talking about the genome of organisms. Specifically, she was working on maize, aka corn, because I don't know if she's bright enough to even know that. How it was arranged and how it could be rearranged when shocks in the to adapt to the environment and deal with stresses from the environment, not toxins, like she goes on to say. It's just the environment itself and how these units of DNA called transposons, which are very specific, can rearrange in the genome, jump into a part of the genome and turn on and turn off genes. And how the genome was very dynamic and active, wrapped around different helper proteins to help it bind and fold and release and expression. I mean, Barbara McClintock was a genius. And then this lady, oh my God, I'm just getting so mad. This lady comes out and says basically that Barbara McClintock saying that, oh yeah, DNA could just basically in a test tube make any sequence that it really wants, just randomly, de novo, just, you can just take a test tube of random DNA from any organism, and if you just shake it hard enough, then it'll form a SARS-CoV-2 genome that you can detect with PCR.
These shocks can be toxins, but can also be from other materials that produce stress in the test tube.
In the test tube. So I'll link you to this here. I believe. Oh, it's way back here. Sorry. Come on. Okay, fine. So I have Barbara McClintock's speech right here. Everyone should go and read it. You know what? I might just read it now because here it talks about the shock. However, the important point here is that Barbara McClintock triggered the activation of DT. DT stands for dotted. It's a locus in the maize genome by crossing in a rearranged chromosome 9. She knew it was capable of introducing a broken chromosome end into a primary endosperm nucleus, causing what McClintock called a genomic shot that set off a nuclear earthquake inside the nucleus, not fucking outside on a test tube.
Can lead to the formation of new genetic sequences.
No, new genetic sequences in the genome because of these rearrangements on transposons, not randomly in a test tube with an unknown mechanism causing development of a known viral genome. It's just, that's not how it works. That's not what Barbara McClintock was saying. This lady should be absolutely goddamn ashamed of herself.
Which were unverifiable, both in vivo and in vitro previously.
Just a minor point is like, even if this was true, what she was saying, that if you just shake the test tube enough, first off, if you just take a test tube and you put it on a vortexer to shake it enough, the genome is going to shred and tear to pieces and is not going to randomly form into new combinations. That's just not how it works. Mostly it just disintegrates. Second is like, it can't be verified. It's like, yeah, it can be. It could be verified through a number of different methods, if that were even true, but it's not true.
So this raises another complicating factor when considering where genetic sequences are actually originating from and how different conditions in life and the lab can change genetic expression.
I mean, the just unbelievable astronomical unlikelihood that a random shock is going to cause a mutation at 3 sites in the genome to be detected by a probed-based PCR 3 times to cause an aberrant PCR reaction in all of the people in the same freaking way is just so phenomenally unlikely. You know, it's just not possible. So there you go.
But even if the PCR test was known to be reliably correlated to a particular organism, something more is required. To prove beyond any doubt that the PCR can truly measure if a person is affected by a disease-causing microorganism, we need to do a blinded experiment. And you guessed it, this hasn't happened today.
Yep, it absolutely has happened, and it's in the exact freaking data that she just is about to produce.
It cannot be emphasized enough how problematic it is when a test has no connection to actual illness. To put it in perspective, there is little doubt that some tests are very useful and have strong relationships to illness. For example, if we have a patient who reports fatigue and a blood test reveals a hemoglobin of 65 grams per litre, we have a very strong indication that they are anemic and need further investigations and treatment. This is not the case with many PCR tests, where a positive result doesn't tell you anything meaningful about the test subject. And as we saw in
it will tell you that the test subject tests positive for the viral samples. And if you look into the details, it will tell you the viral load. And now she brings up Cristiano Ronaldo, who is a soccer player who gives a shit. He's not a goddamn molecular biologist. He's not a goddamn scientist. He was just mad because he broke the quarantine rules that his soccer team put in place, ran off on a trip, came back with a positive result, put in his entire team at risk. You know, he's not the only one on the soccer field who he's not the only one on the soccer field, and he's not the only one on his, you know, with his team that's, you know, not everyone on his team is young, healthy, fit, probably not going to get very bad COVID symptoms if they get any at all. But okay.
2020, even athletes at the top of their game were testing so-called positive with the COVID PCR tests.
Well, he was testing positive and then he didn't get to play. So how are you going to know if he's at the top of his game or not?
To go back to our hemoglobin example, we would not expect Ronaldo to make it through the first 10 minutes of the game if his hemoglobin was 65. So
hemoglobin isn't an infectious disease. So that shows you right there.
I'm sure the soccer star would have much more confidence in this test. PCR tests may be qualitative or quantitative. Essentially, qualitative PCR is the traditional...
No, actually, that's not true. PCR tests can be qualitative as an endpoint PCR, or PCR tests can be quantitative and qualitative, because a quantitative test will give you a qualitative answer when you set the thresholds.
Traditional technique, which is used to decide whether a DNA fragment is present or not, whereas quantitative PCR or qPCR is used to determine how much of the DNA fragment is present in the sample. There has been confusion with regards to this as the product description.
Okay, so now she's going to jump to another test kit. And because this test kit, which is validated by the FDA, doesn't say specifically not for lab use only, it says actually right in here for clinical lab use and for diagnostic lab use. Because of that, she ignores it in the first result where she is trying to say that it's for lab use only. And then she's going to ignore it in the next part where it says that it's very specific. But this is the one that's used for diagnostic determination of SARS-CoV-2 by qPCR. So, and yes, it's intended to be used on a qualitative level to detect it, and it says very specifically right in the protocol at what level to use with it, what machines, what equipment, how to take the samples, and what threshold you set up to take a qualitative measurement with this diagnosis.
Of the RT-qPCR tests for SARS-CoV-2 state. They are qualitative tests, contrary to the fact that the Q in qPCR stands for quantitative. In fact, one of my co-authors, Torsten Engelbrecht, applied pressure to the Charité with regards to the Drosten-Korman PCR test and received the following response. If real-time RT-PCR is involved, to the knowledge of the Charité, in most cases these are limited to qualitative detection.
Right, because at the level that they're testing it at, they're looking for a yes/no answer so that they can get people in and out of the door. And you're not going to run an endpoint PCR on all of those tests because you need to do it on machines. Of course, it has the ability to quantitatively give it a number, but if you just take anyone off the streets and say, hey, this is your CT value, they're not going to know what they're talking about. Perhaps they could have been like, hey, your CT low, your CT high, your CT medium, and that could have given it maybe some context to say like, well, you might be at the end of the infection, or hey, you're at the beginning of the infection. You should really be careful. But the answer either way is you're going to have to quarantine. You're going to have to tell your friends and neighbors and family that you tested and you could become positive.
Clear as mud. In
It is for a conspiracy theorist that hasn't run a PCR in her life.
any case, as I've previously discussed in this video, for some reason the FDA and other health authorities have failed to collect any significant data on PCR cycle thresholds, despite Dr. Fauci saying it was an important value for the clinician to consider. Check out that video for more about cycle thresholds. With regards to SARS-CoV-2, there is an additional issue.
I think I'll probably go over that one with cycle thresholds. I don't remember if she goes over cycle thresholds in this one or not, but this is the best citation that I could find, the study that I could find about cycle thresholds. And that's like these red bars are week 1. This orange bar down here that is barely there is week 2, and then the darker gray bars are week 3 in terms of testing positive for SARS-CoV-2, and then the PCR cycle thresholds are given below it. And as you can see, like, the higher the positive threshold cycle is, which is, by the way, this is goddamn direct evidence that the PCR tests for COVID are spot on with identifying the infectious agent, and that the higher you are, the more likely you are to actually pass on the disease. And you can see at week 1, most of these are above 30 CT cycles. It's not until later weeks that they're lower, and then when they're lower, they get to 0. Most of the ones that are later in the week, if they don't test positive, they don't produce any type of infectious rate. So the way that they did this infectious rate, which may or may not be a 100% adequate representation of how spread is, but they take that material and they spread it on a plate of cells, and even at higher thresholds, past 35, at 35, there's still 3%, 2.7% of the samples will produce a positive result, which means, from their perspective, at a higher threshold, there are still some people who could pass the disease on and be infectious. So that's why the threshold for cycling is so high. It doesn't give you a false positive. It might mean that the chances of you passing it on are, a lot lower, but from a health perspective, you can't tell which type of people are going to pass it on, which type of people are not passing on. So anyway, I'll check out this article here on CT values and cultural virus. It answers a lot of her questions on how CT values relate to infectivity.
And that the target genetic material is for a presumed RNA virus. This means that before starting the actual PCR process, the target RNA must be converted to complementary DNA with the enzyme reverse transcriptase. Professor of Molecular Medicine Stephen A. Buston pointed to the problem that in the course of this conversion process, the amount of DNA obtained with the same RNA-based material can vary widely, even by a factor of 10. So this initial step can also cause variation in whether the subsequent PCR test is considered positive or not.
And it also gives variation in cycle thresholds. That's why health agencies are hesitant to say, hey, you put it at this cycle threshold, this is a positive, this is a negative, when each laboratory has to run their own standards, which is incorporated when you, so if you take the reverse transcriptase and you are running your own standard on that, you're testing for how well that reverse transcriptase is doing at reverse transcribing the RNA molecule into cDNA. So that's why each individual lab has to do that and then determine what their CT values and efficiencies are going to be at that point. Then she goes on to listen at this point very specifically. So she totally bastardizes it right here, where she takes Dr. Buston's work previously about scientific research on RT-PCR and turns it into COVID.
Interestingly, Buston, who helped develop the MIQE guidelines to standardize PCR protocols, had the following to say about COVID-19 PCR tests.
About COVID-19 PCR tests. No, he didn't. This phrase, she pulled it right from his abstract, which is here. Talking the talk, but not walking the walk, about RT qPCR as a research tool, not as a diagnostic tool. We demonstrate that elementary protocol errors, inappropriate data analysis, and inadequate reporting continue to be rife, and conclude that the majority of published RT-PCR results are likely to represent technical noise, because you're dealing with single experiment, single gene, or not single experiments with a couple of genes that you're looking at the expression on, and that people haven't put the rigors into because they're not doing diagnostic tests over millions and millions of people over the globe. This is about single individual research that's being done and not being validated. This is pretty common. It's a pretty common error. And most of his talk right here that she goes into about the RT-PCR step is about reliability between different laboratories, because different laboratories are using different types of RT-PCRs and are not, in fact, doing their validations to make sure that's correct. But each individual laboratory that's doing diagnostic tests for SARS-CoV-2, for COVID-19, are doing their own diagnostic validations, or at least they should be, to set up their parameters. So his entire thing here about how RT-PCR can lead to problems is not to say that it doesn't work. It's to say that you have to set up your parameters correctly. And he's yelling about it, and basically molecular research where you're doing a one-off experiment. You're testing these sets of genes or this one gene, and then you're moving on to the next experiment. You're not doing this over a massive scale, but she's using this inappropriately to suggest that he said this about the RT-qPCR experiments that are happening in individual laboratories, when actually Dr. Buston, in fact, promotes RT-PCR as a solution. It's down here in my notes. Sorry about that. Yeah, here we go. That's the paper, gene quantification. And here's the Eureka, where he developed his own multiplex RT-PCR reaction, and says that it's 100% accurate over 30 samples. Another clinical diagnostic test that she says that there's none of. It's been published in scientific reports, and he specifically says that this is a good way to reliably get test results. But she totally ignores that, or has no clue.
We demonstrate that elementary protocol errors, inappropriate data analysis, and inadequate reporting continue to be rife, and conclude that the majority of published RT-qPCR data are likely to represent technical noise.
Right. So in molecular experiments that they don't set up their controls properly or run a standard curve, it can produce quite a bit of noise, especially when you're comparing apples to oranges between different laboratories and you haven't set up those standards. But he's not talking about diagnostic labs. He's talking about individual research labs.
In other words, a total mess.
And then she extrapolates that totally out of context to mean diagnostic tests in diagnostic laboratories, which is bullshit.
So why are PCR tests being promoted as fit for purpose for COVID-19? It's hard to know all the reasons, but there are huge profits to be made.
But there wouldn't be a profit to be made from an ELISA test or a protein test or any kind of other test. I mean, heck, if they ran it on a mass spec machine, or if they plated it out and did an isolation for every freaking sample, there would not be money to be made on that. So this is a shill argument from a typical conspiracy theorist that are going to talk about shilly stuff.
And advancements of certain agendas. There is also likely to be many conflicts of interest by those with links to political power and public funds for their work.
Because there wouldn't be, if instead of doing this, they set up an ELISA test or a flow cytometry test or anything else. The answer is she would have no answer to what test would be acceptable to her. Everything would be tied to money, because everything is tied to money. We live in a capitalistic society. Even I believe in New Zealand, they still use something called money. So it's just a stupid argument that she repeats again and again.
A viewer sent me an article that was written by a New Zealand scientist called Susie Wiles. She states there was a misinformation video about PCR tests circulating. New Zealand is a small place, so presumably this is one of mine? Unfortunately,
Susie Wiles is a great molecular biologist and microbiologist, and is far, far out of her league. And I agree that she should not have engaged at all. Instead, she goes on in her article, I believe. Boy, I gotta get these organized a little bit better. Sorry, guys. I believe she goes on in her article, which is a really good article, and then just says this is how PCR is a standard good technique. Which, by the way, Dr. Bailey doesn't address at all herself. She doesn't address her arguments herself. She just says, well, why didn't this prominent associate professor in an actual university that has publications and awards and honors not address my conspiracy theory, and instead just said, this is why PCR is a good technique? So,
unfortunately, she doesn't exactly explain which part is wrong, only that it is chock full of false information.
So that's going to be my job, is I'm showing you where this is chock full of misinformation.
Goodness me, what a highly scientific rebuttal.
It wasn't a rebuttal. It's not meant to be a rebuttal. It's not meant to change the people who are infatuated with Dr. Bailey and subscribe to her site, because those people will never change their mind no matter how much information you give them. They don't understand it. It just goes along with their mindset of conspiracy thinking that the COVID-19 is a total hoax. So all she's doing is putting out good information that says good information about how PCR works. And here she'll cherry pick some minor sentences and then try to make some pretty bad arguments, especially if you know the science, but we'll go through it right here.
Firstly, it seems that the spin-off website has
Here's another shill argument. Shill arguments are going to shill.
received $222,000 in funds from Creative New Zealand, which is a division of the New Zealand government.
Doesn't
So
mean the
perhaps the
info's
nature of
bad.
the article is not that surprising. Anyway, let's have a look at where Susie goes astray and how she has cut more than a few corners with her claims. The PCR test looks for specific genetic sequences that are only found in the SARS-CoV-2 virus. As we've discussed, this is a dubious claim, as the virus was never fully isolated and purified prior to the development of the PCR test.
And as we've just discussed, you don't have to isolate and purify the virus in order to get the RNA necessary to first sequence the genome, and then second, develop and validate the PCR test. But it has been isolated and it has been purified in multiple studies across the globe.
It also suggests that we have sequenced the genomes of all significant organisms on the planet and know about every change in genetic expression as per Barbara McClintock's work. Manufacturers of the PCR test certainly don't share Susie's enthusiasm, and in fact
Okay, and then here, now we're on to our fourth cherry-picked SARS-CoV-2 qPCR test kit, which this is, I called them today and asked them, like, what's going on with this test kit? And they're like, oh, this test kit's not available anymore. It's not used for diagnostic purposes. It was used for clinical validation. And they're going to get back to me on what they mean by non-specific interference, but they said it doesn't mean that it's not specific for SARS-CoV-2. Actually, if you look, oops, I don't have it there. Okay. If you look closely at their detection, they say very specifically that it's good for detection against other SARS and MERS, right here, where they say this product provides dual detection of 2 independent genes for COVID-19. They're calling it 19nCoV in single tube. Primers specifically designed for detection of conserved regions of that, respectively, avoiding non-specific interference by SARS 2003 and the bat SARS virus strains. But again, this was used for backup of clinical. It is not intended for diagnostic use, right? So there's clinical work that's being done, but she's throwing shade. Totally avoids all of the FDA and other guidelines to say, like, hey, there are tests out there that are used for research purposes that aren't used for diagnostic. Well, no shit that there are kits out there for research purposes that are not used for diagnostic purposes, instead of going straight to the ones that are used for diagnostic purposes. She cherry picked this one because it says non-specific interference of other viruses, but the tests that have been designed have been checked against other viruses, and they're highly specific.
Disclaimers like, this product is for research use only and is not intended for diagnostic use and may react to other organisms. That makes it very suitable as a screening tool, as it has such a low false positive rate. People are highly unlikely to test positive unless the virus is present in the sample. I can only view this as confused or disingenuous. Where has it been established that it is a suitable screening tool when it wasn't even established as a suitable diagnostic tool?
Yeah, okay, so she is confused and also disingenuous. Here's the genome itself, and here's the genome. Oh, come on. Here's the genome itself. Here's the forward primer. And if I just take that one single forward primer, and I go to NCBI, do a blast search. Yeah, just one of the forward single primers. That's all I need. Now there's probably going to be some non-specific binding to one single primer, but remember, you need 2 primers to bind, and then you need the probe to bind, and those need to bind efficiently enough to adequately amplify the sample to cause the PCR to work, so you can get the CT values. And then even then, after that, you can actually run a DNA melt temp test to see if you likely got the product that you expect to get out of it. And as we can see, out of the one forward primer, 100% of the results are for SARS-CoV-2, showing that this is a very adequately designed primer with 100 hits, and they're all SARS-CoV-2. So that totally makes sense and is highly specific. Do we have all of the organisms of the entire tree of life sequence? No, we don't, but we don't need that because we know that related organisms are going to have similar common genomic sequences. So not only is it going to be specific, and we don't expect some amoeboid, except for in very, very rare cases, to have this transfer from a different organism. We don't even see that in the coronaviruses. Like I said, this PCR is very specific for just this coronavirus.
Cool. The Corman-Drosten paper that triggered the worldwide promotion of COVID PCR tests never established how it should be applied in clinical use.
Yes, it did. Yes, they totally did. If you read the methods of the Corman-Drosten paper, oops, that's, did I put it up? All right, I apologize for that. This is here we go. Corman-Drosten paper. That's right. I brought it up before. You can see that that they give you clinical sampling and how you would use this in a clinical setting. Protocols, application notes, all of that. This is how you would do it in a clinical setting. So yes, it was absolutely set up for doing it in a clinical setting. I don't know what else would you need, Dr. Bailey, in order to make it a clinical test.
And this is one of the many reasons why a consortium of scientists have requested its retraction.
Okay, so Debunk the Funk did look at this consortium of scientists that called for rejection of this paper and said, you know, he goes detail by detail to show it. It's in the show notes, but it boils down very specifically to, like she precludes, is like, oh, well, this doesn't work because worldwide, there's a worldwide misdiagnosis of the infection attributed to SARS-CoV-2, which I believe the editor is considered absolutely outrageous at this point, and have just totally just say, no, that's, I mean, the entire basis of why you want this rejected is because you don't believe that SARS-CoV-2 causes COVID-19, and the evidence for that is just undeniable at this point. I could go through it, but there would be hundreds, if not thousands, of articles to say this is what supports it, including the purification and isolation papers that she says doesn't exist.
It is also unsound medical practice to claim that COVID-19 is diagnosed by a PCR test and that the definition of COVID-19...
Yeah, no, COVID-19 is not diagnosed by a PCR test. It's diagnosed because you have symptoms of a respiratory disease and you're testing positive for PCR. You're testing positive for SARS-CoV-2.
19 is a positive PCR test as a standalone.
No, again, no. It's defined as a respiratory illness, and a lot of people who get it have mild to severe symptoms. Some people have very severe symptoms and death, and that's what defines COVID-19. The PCR test is to help that diagnosis. It's not, the only thing that you're using.
As I've discussed in previous videos, there can be no estimate of false positive rates as there is nothing to compare it to. No gold standard.
Right there. Clinical sensitivity, positives, clinical specificity, 100% negatives. Clinical. Here we go.
Can only be referring to the false positive rate in the lab setting with control samples.
Not clinically specifically. What? It says it right there. So she says that this is not clinically applicable, but these are confirmed cases. I'm just going to go right to the paper. Oh boy. Here we go. So this FIND diagnosis is the one that ran it. And here's how they ran the test. They took samples, clinical samples from the patients, and ran a retrospective study. What needs to happen? Does the laboratory researcher need to be present in the room? Is that what she thinks needs to happen? It's a retrospective study. That means that they took the actual samples and they ran it but guess what? A blinded manner, which she says is what is needed in a blinded manner, with RNA extracted to do the PCR tests. And then they have these examples here. There was a total of 150 samples. That's enough. That's enough to give you a statistical validation on a PCR test. But yet she says there's no clinical test. It says right up on the top, clinical tests.
Not real world application in the community. I've talked about the gross limitations of this in my fact checking video. We know this is true from all the thousands of negative tests we've seen here in New Zealand. This bizarre statement is a complete non sequitur.
Yeah, it's definitely a bizarre statement if you don't know what you're talking about. Because if you have what she's suggesting is that the PCR tests are just first off, she's saying it just tests any microbe that you're coming in contact with, because you're covered in microbes, and PCR is not a statistically or a very specific test according to her. So you're just testing any microbe, which means you're getting tons of false positives. Yet in a country like New Zealand, with hardly any at all, if I believe they even eradicated COVID-19 in their country because of utilizing PCR tests, following the science, not doing what Dr. Bailey suggests, then the test rates, if there's tons of false positives, will not deviate from there being tons of false positives. You would just get lots and lots of positive tests. So the fact that they're running thousands of tests and not finding samples means that there's no false positives to be found, and that the tests are actually very specific. That's not a non sequitur, and it's not confusing to people who know how the tests run.
In no way establishes the suitability of a test. It tells us nothing about the status of the people being tested. There have been no blinded trials to establish the validity of the test for actual illness.
Except for the blinded trials that were just in the tables that you just gave. Those were blinded clinical trials.
Anyway, you can see that she is attempting to build the case.
So first off, she says, oh, there's no clinical diagnostics tests that have been run in the population. Therefore, because this hasn't been run in the population, it doesn't count as a clinical test, even though those samples come from the population and have been run as a clinical test. And then when they say, hey, you know, in a community, there's been thousands and thousands of tests run in a community with no COVID, and they come up with no PCR positives. Then she says, well, then you haven't run the clinical tests to show that that is actually accurate. So it's like when she's like, oh, these clinical tests don't make sense because they're not in the population. And then it's like, these population tests don't make sense because there's no clinical tests. She just flipped herself so that she could make the tests seem not adequate. But in both cases, they make total sense.
Case for PCR tests, but already the foundations are inadequate. Some of the article is fine and talks about cycle thresholds, but then she gets into hypotheticals and a test, trace, isolate mantra public education campaign. Perhaps to her it seems that mass testing can only be beneficial.
Yes, absolutely. It's definitely beneficial because it allows you to find hotspots, and then when you're testing those hotspots, you trace those individuals and you isolate to find new variants that could be highly problematic. As we can see, you know, I like the people who are like, just have this conflict of they can't determine if this is a bioweapon that was engineered by the Chinese government to attack the United States, or I mean, to attack the globe. But also the PCR tests are inadequate and the numbers are inflated and the deaths aren't even really that bad. I mean, one of those will be true, but not both of those. And the other thing is that at the same time, it's like I lost my train of thought. Sorry about that. Let's just have her go ahead.
But this ignores the huge costs that include financial costs. An Official Information Act request in New Zealand, a country of 5 million people, revealed $74 million in PCR, swab and lab costs alone over a mere 6-week period.
I mean, but shutting down your economy has huge ramifications and way larger costs than that. I don't know what the GDP of New Zealand is, but I can guarantee you it's much larger than $74 million.
The total cost will be far in excess of this, and be psychological and social costs to people who believe that they are actually ill and isolate because of a positive test result.
The psychological and social costs that I've seen firsthand in a country Spoiler alert, I'm from the United States, that is suffering tremendous loss. The consequences of people like losing people, those psychological consequences are far, far higher than, oh, you have to stay home, you can't go to work.
However, most conspicuous by its absence in the article is where it was established that positive PCR test results have anything to do with illness.
Yep, here we go. Nature, communications, SARS-CoV-2 viral load is associated with increased disease severity and mortality. I guess if she would look at this, she would find it, but yep, if you're in worse shape in hospitalizations, you have a higher viral capacity. It shows it in several spots right here in terms of your the viral load that you have and the significance of death much higher if you have much higher viral loads. This counters exactly what she says.
She completely misses this crucial point, and this is not surprising
because she didn't address it. She was only addressing the PCR and why it's effective as a screening method and a diagnostic method, not how it's related to morbidity. Otherwise, she would have quoted the Nature article that shows it clearly has a close correlation to morbidity.
Because, as I discussed in my video, What is a COVID-19 case? It has never been established. As my co-author and seasoned campaigner Klaus Kernlein would say, she has been swept up in a PCR pandemic. Unfortunately, scientists like Susie seem to have become detached from the very nature of human health.
Yeah, look in a mirror much.
As they focus on molecular test results and top-down political policies. Interestingly, some of the authorities that heavily promoted the COVID PCR tests are now softening their positions, with a European court declaring the tests not fit for purpose and potential lawsuits brewing. Last month, the WHO
No, they're not saying that it's not fit for purpose. There's some lawsuits about keeping people in quarantine because they can't determine if they have a low viral load or not. But like I said, from the health perspective, even if you have a low viral load, while some people have low viral load, can still pass that on. And from a health perspective, you can't separate who those people are or will not be. It doesn't mean that the PCR tests themselves don't work or they don't function. And then now she's going into the WHO notification letter that basically is telling lab users, hey, make sure you take into consideration the laboratory manuals that state how to run these PCR tests. It's just a reminder. They come out with these kind of things all the time. You just don't know it because you're not a laboratory technician running these types of tests. They're not throwing shade or doubt into any of the findings. They're just saying, follow the PCR manuals.
Quietly issued a statement suggesting more caution with so-called positive tests.
Healthcare providers are encouraged to take into consideration testing results along with clinical signs and symptoms, confirmed status of any contacts. Confirmed status of any contacts. That does not say exactly what she said.
Could this be the beginning of a gradual backdown from unsound science?
If it was, you would abandon your YouTube videos.
Over a decade before PCR tests appeared on the scene, Nobel laureate for medicine, Sir Frank McFarlane Burnett, made a prescient warning. By the beginning of the 1970s, he had become very skeptical about the usefulness of molecular biology, especially because of the impossible complexity of living structures, and particularly of the informational machinery of the cell. Certainly molecular biologists are rightly proud of their achievements and equally rightly feel that they have won the right to go on with their research. But their money comes from politicians, bankers, foundations who are not capable of recognising the nature of a scientist's attitude to science and who still feel, as I felt myself 30 years ago, that medical research is concerned only in preventing or curing human disease. So our scientists say what is expected of them. Their grants are renewed, and both sides are uneasily aware that it has all been a dishonest piece of play acting. But then most public functions are.
Hilariously wrong. He was known for being good scientist about immunology. And then when molecular biology came along, he hilariously made this statement. And then PCR came out almost like, less than a decade after he said that. And then his entire concept is like, molecular biology will be dead within another couple of years because the applications are too complex. PCR comes out, shows that you can do a diagnostic test using these molecular practices, which again, she ignores. She ignores the fact that PCR was designed first as a diagnostic test for a disease, and then hilariously quotes the guy who says that molecular biology is a nonsense field that will very quickly dissipate. And, 30, 40, 50 years later, molecular biology is on the cusp of the bleeding edge. We've just invented CRISPR. We've just discovered how to use mRNA vaccines. That'll probably lead to a cure to cancer or any number of applications. Molecular biology is on a roll. This virus would be far worse if it wasn't for the fact that right away, immediately, the viral sequence of the genome was sequenced, and agencies around the world could pick up and make their own PCR tests immediately. And because we've had 35 years of knowing how PCR tests work, we came up with a phenomenal test. These tests are really great. And the countries that put the best amount of effort into their PCR tests had the best results, including Korea, New Zealand, and Australia. These places have had phenomenally low deaths caused to COVID and have phenomenally low cases, which have allowed their countries to open up. Implementation of tests here in the States have been slow. It was bogged down because we had contamination, because we know how to run PCR and because we know controls. We know that contamination is an issue, and we're able to detect that contamination was an issue so that we didn't get aberrant results. But then that slowed down our ability to adequately test for it and figure out who had it and who needed to isolate. And it was a disaster, and the virus ran over our country. And now we've had up to, probably even more than 500,000 people have died. It's a total tragedy. And listening to people like this is just grinding, and a real tedious. It's really tedious.
As a quick comment, I can also say that when PCR tests are used out of context, they can also appear to provide evidence of a new strain of a virus.
That's not how strains are done or isolated. The PCR tests don't point you to that. The isolation and the sequencing points you to that.
Again, this indirect method does not suffice, and full evidence of the virus needs to be carried out with formal purification and isolation, as well as the characterization of the full genome and shell. And if there is evidence of any new strain, we still need evidence that it poses any threat to humans.
Yeah, here is the evidence. The CDC is tracking that evidence continuously because it's a problem. Everybody knows it's going to be a problem. And oh gosh. Oh, here we go. Sorry about that. Yeah, this is the isolation screening sequence. Variant detection is here. Apologize for that. Okay, here are all the variants that have been detected. And as you can see, the variation is across the entire genome. This is the entire genome, all 29,000 nucleotides, and they're tracking all of these variants across all of this time and all over the states in the world. This is CDC data. You can't get this off of a PCR test, like she says, that just tests specific regions, because those are quick tests. So what they're doing is they're looking at regions that are hotspots. Then they're taking isolates from those. So you could take a PCR test at the same time you're taking a sample that can be isolated later. So they're randomly taking samples and then doing these isolations. They're not developing their understanding of variants off of the PCR tests, although that is another way that you could do it. If you know which area to target, you can do it that way with melt temps through real-time PCR. Of course, you would know absolutely nothing about how to do that because this is not how she operates. But yeah, no, the variants are not being detected through PCR. They're being isolated, purified, and then sequenced. It's just they're not isolated and purified the way that she deems that they need to be isolated and purified, because we have advanced techniques that allow us to just take the viral genomes of these cultured cells and then we can do genomic analysis on that. She's so far behind.
As its mere detection is no reason to cause panic. In summary, I'm not saying that there is no use for PCR tests. They are an incredible tool of human discovery and have a vital role in scientific research, as well as genetic and forensic medicine. However, their use with regards to infection diagnostics has generally been overplayed in recent years.
So hilariously, in her next video, which was part 1 of this one, she lionizes Carrie Mullis after she just got done saying that forensics is the great purpose for PCR. Carrie Mullis famously went almost on the stand in the O.J. Simpson trial to say that PCR doesn't work in forensics and doesn't detect anything. That's the problem, is if you go with Mullis's logic, you can't rely on PCR for a test for anything, which is what he wanted it to be, because he wanted to throw shade in the corporation and the industry that basically shut him out and made a bunch of money on a very effective technique that he basically developed. But then they had to take it and develop it further into probed assays and assays to test for sickle cell anemia. It's just, PCR is a very simple, very flexible method that's used in the totality of science. So anyway, just in summary, right here, she leaves out specificity of PCRs with probes and multiplexing. That shows how specific the PCR reaction can actually be in the genome. It's very specific on 3 different levels in a single PCR reaction, and then you have 3 PCR reactions on top of that. She confuses DNA sequencing with gene expression and absolutely butchers Barbara McClintock's work on transposable elements and the flexibility of the genome inside the nucleus of living organisms to react to their environment, to mean basically that DNA can develop all sorts of random aberrant sequences. It's just not true. And it just shows you like how she's taking these characters through history and sprinkling them into her notions so that she seems smarter. Bailey cast doubt on PCR methods by jumping to random PCR kits that are not used for diagnostic settings. Some of them are approved for diagnostic settings, but the manufacturer themselves don't do the approval process. The FDA does the approval process, or the other regulatory agencies all over the world do the approval process. So they don't put on there that this is for diagnostic purposes. They haven't been the ones that submit the paperwork and go through the tests. The FDA does that through their emergency use authorization, but most diagnostic labs use their own validation process and their own set of validated FDA protocols in order to do clinical testing. So it's just throw shade, throw doubt on the whole thing.
And I suspect 2020 will be viewed as the year in which they went completely out of control.
Nope. PCR testing for diagnostic purposes on disease will forever in the future be used I doubt there will ever be a more sophisticated method that is that rapid for developing, and she's just wrong. She is just wrong. So I see a lot of other things coming out. Of course, the Organic Consumers Association is now promoting Ronnie Cummings and Dr. Merkele's new book on the truth about COVID-19. Anytime I see the truth about something now, it seems like the opposite. That's a bunch of misinformation. And I said it's like the RNA virus was indeed engineered in a lab. I think I'm going to go into this because I'm seeing a lot of lab engineered origin stories, and they're so all over the place, and they are just like pointing at it's almost like the old timey UFO, like, you know, this is definitely evidence of UFOs. This is definitely evidence of Bigfoot. They have no idea what they're doing. And like I said, it's a bioengineered weapon from China that is used by the global elite, and yet PCR tests are all wrong, case counts are all wrong, morbidity is all wrong, and safety efficacy is all wrong. And it's like, well, pick a lane, man. It's either this is the worst bioweapon ever, or all of it's wrong. Either lane you're in, you're wrong, but you don't even see the contrast between it's a terrible virus that's actually not doing anything. So again, I'm producing these. They are very rough, and I'm just getting them out there so that people have the information. And I'm pretty sure it's going to get a lot of negatives because it's not very well cut, but I don't care.
Your boos mean nothing. I've seen what makes you cheer.
Yep. So, all right, have a good one. We'll see. Hopefully some of the bigger ones like, Debunk the Funk or Potholer 54, or maybe even Thunderfoot can pick up on this and produce it into a video that would be much better for the general lay audience. This is more for the skeptics who just need like a refresher on how wrong she is and where the evidence actually lies on countering her nonsense. So, all right, have a good one.